So friends, before we move on to some clinical correlation of this particular topic that we have just discussed, I want to deal with few important terms which are the recent terms, and they require your attention because recent advances are of utmost importance. There is something known as Emperieolesis. Now, this term is not something that is new to the pathologists, but definitely you can say it is a new favorite with the examiners. We just spoke about phagocytosis. Phagocytosis was that there was one cell which was eating up a bacteria or any organism and killing it. Emperieolesis is also something similar. What is it? That is, if this is one particular cell, right, and this is, say, another cell. Now, just for diagrammatic and for understanding purpose, you can say this is the bigger cell and there is one smaller cell, and the bigger cell will eat up the smaller cell. The smaller cell will be present inside the bigger cell now. But what is different? Why am I just not calling it phagocytosis when one thing is eating up the other? If you notice, over here there is no killing that is taking place. Over here you can say that there is one intact cell. There is one intact cell inside another. There is one intact cell inside another. There is no killing happening in this process. When one cell, one intact cell, eats up another intact cell and none of the cells die, there is no killing, that process is known as Emperieolesis. So do not confuse Emperieolesis with phagocytosis. Phagocytosis is a killing mechanism. Emperieolesis does not involve killing. What are the examples is what is asked. What are the examples where this Emperieolesis can be seen? So the example includes, now we just need to write the same term again, Emperieolesis. And if you see, I have focused on two terms more or two letters more. These include M and L. Why have I focused on these? Because this will be the mnemonic that you will be following. Emperieolesis is seen in a condition known as MDS or myelodysplastic syndrome. This is one syndrome that we will study eventually in hematology, myelodysplastic syndrome, and this syndrome is showing Emperieolesis. That is, what cell is eating? Remember the megakaryocyte. The megakaryocyte engulfs other cells. What is a megakaryocyte, students? Megakaryocyte is the precursor of a platelet. It is a precursor of a platelet, and megakaryocyte in MDS, in myelodysplastic syndrome, starts eating up other cells. So that is the first example with M. There is another example with M, that is multiple sclerosis. That is multiple sclerosis. Right? Now, M for MDS, M for multiple sclerosis. In multiple sclerosis, the astrocytes engulf the lymphocytes. Astrocytes which are present in the brain, they will engulf which cell? The lymphocyte. Is there any killing happening? No. So this is an example of one cell taking up another, that is Emperieolesis. The next letter that I told you to focus on was L, because Emperieolesis is also seen in lymphocytic leukemia, and to be more precise, it is seen in chronic lymphocytic leukemia, or I can say CLL. CLL, that is chronic lymphocytic leukemia. So two letters, M and L. M for MDS, where M, the megakaryocyte, will show. M for multiple sclerosis. L for lymphocytic leukemia. Which lymphocytic leukemia? Chronic lymphocytic leukemia, or CLL. So this is one point that I wish you to differentiate, be able to differentiate from phagocytosis. Now, what is age-old is phagocytosis? Ever since pathology has been read, phagocytosis is being read. a newer form of killing has come up, which has now become a very favorite with the examiners. And that particular form of killing is something known as nets. Now it is not the image that is shown over here. It is actually sounds similar but is different. What is the full form? If you know, you already know the entire mechanism of what is happening. So this stands for neutrophil extracellular traps. So nets, N-E-T-S, neutrophil extracellular traps, right? So if I ask you, what is the cell that will be involved in this type of killing? You know the answer. It will be a neutrophil. If I ask you, will this killing be intracellular or extracellular? Now again you know it will be extracellular. And if I ask you, what is the mechanism of the killing? You know there is formation of some trap. So remember, this is a special form of killing. This is a special form of killing which will be carried out by the neutrophils, outside the neutrophils, extracellular. Now what exactly is happening? Nets is something that comes into play when the body is in sepsis. That is a severe bacterial infection, body is in sepsis. In sepsis, there is activation of platelets. And these platelets, when they get activated, they are going to help in the mechanism of nets. They are going to activate this mechanism. So when the body is in sepsis, platelets get activated. Platelet activation eventually results in the formation of these nets. Now what exactly is happening over here? Let's try to understand this with the help of a diagrammatic representation. Now students, for example, I've taken up a particular cell that you can see. This is I hope all of you can recognize 3 nuclear lobes. It is a neutrophil. What you see that maybe surrounding it we have either the bacteria or a yeast or a protozoa or any organism that has come in, right? Any organism, any infectious agent. How will this killing happen? The first thing that you notice over here, there were 3 lobes in this neutrophil. All the chromatin has dissolved. It has opened up. All the chromatin in the nuclei, instead of being compact as 3 lobes, all of it has dispersed out, right? After dispersing out, what is the next thing that has happened? All of the chromatin has been sent out. Like these fibers, all of the chromatin has been sent out of the cell. What is this chromatin doing? It is forming a trap. So we've come to the 3 terms. Neutrophil started it. A trap was formed and it was formed outside the cell, extracellular. So neutrophil extracellular traps. Whom are we trying to trap over here? Whatever bacteria, virus, protozoa was causing infection in our body, the neutrophil chromatin will trap it. If you've understood this, let's try to now put this down in words and understand this in slightly more detail. So for example, there is a neutrophil, right? Neutrophil is going to give out chromatin fibers. So chromatin fibers are given out or given extracellularly, you can say. That would be a better term, extracellularly. First question that arises, which amino acid will help in doing this to the chromatin? Which amino acid will help in nets or will help in giving out the chromatin? And remember, the answer is arginine. This is one of the most recent questions that has come up 2017 onwards. That is, arginine is the amino acid which is going to help in the neutrophil giving out all of its chromatin. The chromatin goes out. As soon as the chromatin goes out, it is going to result in the formation of a trap. Whom will it want to trap? The bacteria or any organism that is present. But just by trapping the organism, can we kill the organism? No. what instead will happen? The chromatin trapped the entire bacteria, right? After that, the neutrophil is going to now release all its enzymes. The neutrophil now releases enzymes inside the trap. And once it releases enzymes inside the trap, this will definitely result in killing. This will result in killing. So there is a neutrophil. Because of amino acid arginine, all the chromatin goes out. All the chromatin goes out, it forms a trap. In that trap, all the enzymes are released from the neutrophil. The organism dies. But please note one very important mechanism over here. If a cell leaves all of its chromatin, all the nucleus has gone out, all the nuclear content, chromatin that it had has gone out. Can a cell survive without its nucleus? No. So in the process of killing the particular bacteria, the neutrophil also dies. The neutrophil also dies at the end of killing. So I can say that this is nothing but an example of a beneficial suicide. That is the other term that is given to it. Beneficial suicide. That is, the neutrophils themselves will die. So we've come across 2-3 very important questions over here. First, the chromatin dispersion happens because of arginine. In the end, the neutrophil will die. So that is a kind of a beneficial suicide. These are going to constitute a trap, and finally the organism will die. Now what we need to know is a recent image-based question that has come up, and you need to focus on that. Now this is an image showing a neutrophil extracellular trap. What do you mean by this? So first I would want you to focus on this image above. This particular image. Can you see that there are these 3 cells over here? What you see in red is the nucleus, and what you see in green over here, what you see in green is the cytoplasm. So this is actually a neutrophil. Red part is the nucleus, green is the cytoplasm. Now come to the one that is beneath it. If you see, the green part remains intact over here. What has come out of the cell? All the red part has come out. So this is nothing but a neutrophil extracellular trap, right? All the nuclear chromatin, the red was the nuclear chromatin, all of it has been sent out and is going to trap the organism. That is a neutrophil extracellular trap. So this is one of the more recent forms of killing which you need to differentiate from phagocytosis, and 1 or 2 very important differences are phagocytosis. Remember, phagocytosis, if you want to differentiate from neutrophil extracellular traps. So the first point is that phagocytosis was intracellular. All the killing was happening inside the cell, and NETs, as the name suggests, is extracellular. Also another difference that lies is, the second point, is that at the end, what happens at the end? Bacteria will be killed in both, right? But in phagocytosis, the cell remains alive. But in neutrophil extracellular trap, I just told you that the cell will die, and that is known as a beneficial suicide. That is known as a beneficial suicide. So these 2 important differences you must know between phagocytosis and NETs. And to repeat again, Emperieolesis and NETs, these are 2 important topics that have come up now, which are becoming favorite with the examiners. So we need to focus on these more for the upcoming exams.